Howdy, my name is Angelica Ramirez, and today I'll be presenting immunologic tolerance, autoimmunity, and hypersensitivity. So, how do the immune system maintain unresponsiveness to self antigens? First off, we have central T lymphocyte tolerance, which are when the T cells react strongly to self antigens are eliminated in the thymus. Since this process is called negative selection and ensures that only T cells that recognize non self antigens are allowed to mature. The mechanism of this is T lymphocyte anergy. When the T cells recognize antigens without co-stimulation, the TCR loses its ability to transmit activating signals. CTLA4 and PD1 are very important as they mediate the suppressive function of regulatory T cells and terminate the responses of T cells to self antigens. Central B lymphocyte tolerance has three mechanisms as well. Receptor editing re-expresses RAG genes, resume immunoglobulin light chain gene recombination, and express a new immunoglobulin light chain. Deletion is when receptor editing fails. Immature B cells die by apoptosis. And anergy is when antigen receptor expression is reduced and the cells become unresponsive. This is kind of like a picture, so you can kind of see the visuals of both central B and T lymphocyte tolerance. Peripheral T lymphocyte tolerance is when autoreactive cells escape and enter the periphery after central tolerance mechanism. Peripheral tolerance mechanisms act outside of the primary lymphoid organs to prevent these cells from causing harm. Anergy and deletion are also in mechanisms of the peripheral tolerance, but regulatory T cells are the T cells that suppress immune responses, maintain tolerance, and prevent autoimmune responses. Peripheral B lymphocyte tolerance has anergy, deletion, and regulation by inhibitory receptors as their mechanisms. Again, this is another picture of peripheral B and T lymphocyte tolerance. So, what are the factors that may contribute to the loss of self tolerance and the development of autoimmunity? We have two factors: genetic and environmental. Genetic are when polymorphisms in non HLA genes, which is the human MHC, are associated with various autoimmune diseases that may contribute to the failure of self tolerance or abnormal activation of lymphocytes. Diseases like Crohn's disease, type 1 diabetes are autoimmune diseases that are contributed to by genes. The environmental stimuli is when normally mature T cells with self antigens are presented by resting tissue APCs and they result in peripheral tolerance. However, microbes can activate APCs to express co-stimulators, and when APCs present self antigens, the specific T cells are activated instead of being tolerant. Some microbial antigens may cross-react with self-antigens mimicry, and immune responses are initiated by the microbes may come directed at cells and tissues. What are the mechanisms of different types of hypersensitivity? We have four types of hypersensitivity. Type 1 is a type of pathologic reaction that is caused by the release of mediators from mast cells. So when an allergen enters the body, the activation of immunoglobulin E antibodies is activated and binds to mast cells and basophils. Subsequent exposure to allergen cross-links the immunoglobulin E antibodies, leading to mast cell degranulation and release of histamine and other inflammatory mediators. Examples of type 1 hypersensitivity are allergies, anaphylaxis, and hay fever. Type 2 hypersensitivity are when IgM or IgG antibodies bind to antigens on the surface of cells or tissues, leading to cell destruction by complement activation or phagocytosis. Antibodies can target self-antigens or foreign antigens on cells. Example of a type 2 hypersensitivity are hyperthyroidism, rheumatic fever, and pernicious anemia. Okay, this is kind of like a visual of type 1 and type 2 hypersensitivity mechanism. Type 3 hypersensitivity is when a soluble antigen-antibody complex is formed and deposited in tissues, activating complement and attracting neutrophils that lead to tissue damage and inflammation. An example of this is serum sickness, Arthus reaction, and polyarteritis nodosa. Type 4 hypersensitivity involves the activation of T cells, which are CD4 and CD8 T cells, in response to antigens, resulting in the release of cytokines, recruitment of inflammatory cells, leading to tissue damage. Usually takes about 24 to 72 hours to develop after exposure to the antigen. And some examples of this are poison ivy, tuberculin skin test reaction, and even graft rejection. Again, this is another picture or visual of type 3 and 4 hypersensitivity mechanisms.